Ayro, can you hear us? Or Francesca?
No,
Navidad.
Trinidad. Is error online?
Bueno, pero algo así,
There are some people here who do not study biophysics, so I will start with a little introduction about why this is an important problem and what are proteins. So, there are many things that we associate with life. If you think about something that is alive, it can move. It can sort of sense information, it can understand this talk because of our ability to process information. All things that are alive also metabolize energy, they grow, they reproduce. And the only reason why all of this happens is because of the action of molecules called proteins in all our body. Of course, we know about proteins as something we eat, but the reason we eat them is because the building blocks are then used to make new proteins that do all of these things. For example, when we move around, proteins are what actually allow our muscles to contract relative to each other. We have ion channels in the brain that conduct electricity. And so proteins kind of do all these things. And so from our day-to-day intuition, we know about the importance of structure, right? How things are arranged in space. So if you think about a macroscopic structure, like the famous Golden Gate Bridge, which, I'm lucky I can see most days in San Francisco, except some days the fog comes in and then you can't see it so well. But most days you can see this bridge. And the reason that it can carry so much traffic is because all the posts are arranged in exactly the right way. The cables are arranged in such a way that they can carry this immense load. And these same principles are at play in proteins as well, but now we're talking about the arrangement of atoms in space rather than about posts and cables. And so the question that I try to understand is, okay, and the another important corollary is, when a structure is not built correctly, it collapses, right? A bridge can collapse, for example. And it turns out that this same principle holds when it comes to proteins. So when proteins don't quite acquire their correct three-dimensional shape, they do not function and have been implicated in many diseases. So there are many diseases that are called protein misfolding diseases, where the arrangement of atoms is not quite correct in space. So the most well-known case is actually Alzheimer's disease. In Alzheimer's, one of the driving events is the misfolding of this protein called amyloid beta, which normally stays very well in solution. So of course our cells are filled with water and proteins need to be well dissolved in water. But when this protein called amyloid beta does not quite get its correct three-dimensional arrangement, different molecules will start to stick to one another and form toxic oligomers, which can actually damage a cell. Or these are kind of deposits of proteins. And if you actually dissect the brain of somebody, post-mortem of somebody who died from Alzheimer's, you will see these deposits of proteins because of the misfolding. And the same is true in Parkinson's, ALS, as well as certain types of cancer, cystic fibrosis, and many other diseases, actually, amyloidosis, various others. So given the importance of this process, it's very important then to understand what ensures that in healthy, normal cells, proteins get the structure correct, and then that can tell us how to intervene when this process goes wrong in all of these diseases. And one point I want to make is that the field of protein, just like everything we all work in has been transformed by artificial intelligence, right? We all use now ChatGPT. It's such a big part of our lives now. And it turns out that the field of protein folding has also seen a huge role of tools from AI, from artificial intelligence, including the well-known AlphaFold algorithm. But it turns out that this question of how proteins get their structure is not addressed by AlphaFold, because AlphaFold, all it does is it predicts for you the native structure of a protein with remarkably high accuracy, meaning the structure that it attains at the end of the day when everything goes correctly. But it does not tell you about how that structure is attained in the very complex environment of the cell, and by corollary, how it goes wrong, how this process goes wrong. So again, a little bit of introduction for those who don't think about proteins every day. Proteins, of course, are made of amino acids that are one in sequence with each other. They're chemically bonded to each other. And at every position in the protein, there can be 20 different amino acids with distinct physical and chemical properties, right? So for example, there might be pairs of amino acids with opposite charge that will attract each other. Some amino acids are greasy, basically, so they try to avoid the water that surrounds all proteins. And somehow, and the genes are DNA, and then via the intermediary of the mRNA is what actually instructs in what sequence these building blocks need to be strung together. And then somehow, and this is a big question mark of my talk, somehow all of these interactions, as well as other factors in the cell, lead you to go from a one-dimensional string of information, a sequence of amino acids, into a very precise three-dimensional shape. And that is the question that I'm trying to understand how it works. So let's think about how this might happen. What we see is that in this string of amino acids, there are, as I mentioned, different types of physical and chemical properties. And you could imagine that maybe those interactions are all that's necessary to drive this chain into the right shape, right? Maybe just because different ones will attract due to charge, different ones will want to bury themselves in the core, maybe those interactions are sufficient, right? And this is the simplest hypothesis for protein folding known as Anfinson's dogma, where if you take a protein that's correctly folded, put it in conditions where it loses its structure. For example, if you raise the temperature, that actually causes proteins to do something called unfolding or denaturation, and then put it back under conditions that are more hospitable, the theory is that the protein should reattain its native structure or refold, right? And this has been shown to be true for a number of proteins. Christian Amundsen, who got a Nobel Prize in biochemistry, was the first to demonstrate that this, at least in some cases, is how proteins fold. And the analogy is that folding is an energetically favorable process. So the analogy would be if you place a soccer ball in where we were this weekend, in the top of the Andes, and then you just let it start to roll however it wants to go. Eventually, it might end up down here in Santiago, right? Because the gravity is the equivalent of the interactions between the amino acids of the protein. And just by the energetics, the lowest energy place would be for the ball to land down here in Santiago, right? So for some proteins, the interactions between amino acids play the role of gravity and cause folding into the correct structure in and of themselves. But we're starting to realize that that's not necessarily true in general. There are actually some proteins that are larger, meaning that the number of amino acids in the chain might be, instead of like 100, might be 200, 300, or even more than that. Where for those proteins, if you start them off, if you sort of take the protein, purify it from cells, so you have a solution just of that protein, and then again, heat it up or put it in denaturant or some condition where it loses its structure, and then try and put it back in conditions that are more favorable for the structure to form, it will not get the structure correct. It will actually end up in some different structure, is called a misfolded structure. And these misfolded structures sometimes can then go on to do this process of aggregation where the molecules stick to each other. This is something that's been linked to the diseases I mentioned before. So, and in fact, there's some recent work that has shown that a significant subset of proteins are actually prone to misfolding when you try and unfold them and refold them in this way, telling you that this analogy that Santiago is like the correct structure of the protein might not be correct. Maybe if you start them at the top of the hill, they will end up somewhere completely different. So how do you resolve this problem? Because our proteins need to fold correctly, right, in order to function. Well, the simplification we were making here is we were sort of assuming that all you need is the protein by itself, but that's not how the cell works. In the cell, there's actually a lot of machines that help proteins with this process of folding. And these can very much shape these mechanisms. And so, of course, those are going to play a role for these proteins that do not fold autonomously by themselves. So one of the most important sort of cellular mechanisms that is involved in folding is protein translation. So we have all of our proteins are synthesized on the ribosome, which is what reads the information, our genetic information, in the form of mRNA, which then tells the ribosome in what sequence to add amino acids. So it's kind of the factory where proteins grow. And it turns out that as a protein grows, it can already start to fold itself up, at least in some cases. And you can already start to see that this is going to cause folding to happen in a very different way than in the simple scenario I showed you before. Right? The simple scenario was just you have a complete protein already there, all the amino acids already strung together. You unfold it and then try and fold it again. Here, the number of amino acids present in the chain changes with time, right? Because as the ribosome adds more and more amino acids, the interactions that are available, the information will continuously change. So the analogy is, again, you put the ball at the top of the Andes, but now as it starts to roll down, there's a big storm at the same time that creates erosion. So that as the ball is rolling, maybe new valleys will continuously form, right? New areas where it might be able to roll. And depending, of course, on how quick is this erosion relative to how quickly the ball is rolling, that will affect whether it goes into one of the new valleys that forms or whether it eventually, again, makes it down to Santiago, right? So the idea is that this is what we call an out of equilibrium process because the landscape that the protein is experiencing changes dynamically as more and more amino acids are added. And this can naturally affect in very important ways how a protein folds. And in fact, it's been shown that there are certain proteins where if you do the experiment from the last slide, unfold them and refold them, they don't fold correctly. But with this out of equilibrium process of translation, where you're kind of always creating these new valleys in the landscape, they do actually fold correctly. And that's just with translation by itself. You don't need chaperones, you don't need anything else, at least for some of these cases. Just the fact that the dynamic energy landscape changes already helps them fold more accurately. And there's actually evidence from evolution that folding on the ribosome in this way is something very helpful and that it's conserved across evolution. And typically when something is conserved, if you see it in a lot of organisms, it means it must be important. And the way that this manifests itself is that it turns out there is a way to sort of program the speed at which the ribosome adds amino acids. So it's a little bit analogous to programming the speed at which, say, a storm maybe forms new valleys during the erosion process, right? And this is what's called codon usage. So you can have what are called rare codons in a protein sequence that cause a pause in the process of translation at certain points. And it turns out that there are some organisms where you have many of these rare codons in a row in the protein sequence, meaning that the ribosome will stop at one specific place, potentially to give time for the protein to fold. And in fact, some theoretical work has suggested there's actually a very strong correlation between pausing positions and folding events. So it's as if, I don't know, you have one particular place where maybe there's one area in the path of the ball that is very prone to erosion, and you always see erosion at that particular place so that maybe it will guide the path of the ball very. And of course, and I think this is something that Ito's lab will especially kind of be able to appreciate, that events, now it works. So events on the ribosome are very important for other cellular processes, right? So chaperones and including, translocation machinery will recognize nascent chains as they're first being made on the ribosome. And a lot of this will actually depend on the conformation of the nascent chain as well. So all of this tells you that what happens on the ribosome is extremely important for the fate of all of our proteins, right? And the fate of our proteins is very important, of course, for their ability to do all the functions that we talked about before. So it would be really great if we could actually see this. This is just a cartoon I made. It has no basis in reality what I drew here, but it would be great if we could actually see what's going on there, right? And so what I'm going to tell you now is that I've done some work in the Marcus and Bustamante labs, as well as some simulation work during my PhD with Eugene Shaknovich that actually lets you solve this big gap in the field, where we now have ways to actually see what is happening to a protein as it's coming out of the ribosome and starting to fold itself up when it's actively being translated. And so a little summary again, I will tell you a little bit about both some experimental work as well as some theory and simulation work. Probably most of it will be this more recent experimental work that I'm doing now as a postdoc. And I'll kind of tell you about how combining techniques from experiment with techniques from simulation, including approaches, not necessarily just AlphaFold, but more physics-based approaches, is actually a really powerful way to learn about, in a high level of detail, how folding happens in cellular-like conditions. So I'll start off by talking about this experimental work. So now I'll get a little bit more into the details of how some of my experiments work, and then we'll talk about what we learned from them. So how can we watch a protein as it starts to fold? We have actually a very good way of doing this, not on the ribosome. In that very first type of scenario I told you about, where you have just a purified protein and you can unfold it and refold it, we have a way of figuring out how different parts of the protein fold and in what sequence. And that's called pulsed hydrogen-deuterium exchange mass spectrometry. And I'll tell some of you might know about it, but just for those who don't, I'll tell you a little bit about how it works. Where let's imagine you have a solution of proteins that starts off in an unfolded state. Maybe you took the molecules and you heated them up, or you put them in urea or something that will make them unfold. And then at time 0, you put it in conditions where folding will become possible. And then let's say at time t, you want to ask the question, what parts of the protein already started to fold and which ones have not folded yet? What you do is you take a little sample, a little aliquot from that reaction, and you dilute it in a different type of solvent called deuterium D2O, where if you think about water, in water, normally the protein is in water, and water has 2 hydrogens and 1 oxygen, and the hydrogen is just a proton. It's a sort of very light element. This is just a different form of water, but the hydrogen now has a proton and a neutron instead of just being a proton. So it's a little bit heavier. And why we do this is because this turns out that it serves a little bit of as a sticker. It tells you what parts of the protein were not yet folded and which ones actually had started to fold. And the way that works is across the chain of a protein, you have amide groups, a type of chemical group where the hydrogen on the amide will readily exchange with a deuteron, meaning those parts of the protein locally will become a little bit heavier, only though if those parts of the protein were not yet folded. If the parts started to form stable secondary structure, means stable hydrogen bonds, they will resist this exchange. And so what you could do is at different times, you label your protein with deuterium, and then you can actually analyze which parts of the protein took up the deuterium and which ones didn't. By quenching this exchange reaction, you actually have to acidify the sample so that this exchange between hydrogen and deuterium stops. And then you add a protease to this protein, which is a type of enzyme that digests the protein into these little chunks called peptides. And you can then inject these into a liquid chromatography mass spectrometer, where this is sort of a fancy equipment where what it does is it's like a scale, a weighing scale, but for very small things. It tells you how heavy these peptides are. And what you'll often see is you'll see what I'm showing here is how probable it is that you see kind of different masses, right? And oftentimes when you do this experiment, you see these true distributions like this, where one distribution is the molecules that have heavier mass. When they have heavier mass, what that means is that that particular peptide has deuterium, meaning that when this labeling was done, the part of the protein from which this region came, which is this yellow and red part, was not yet folded and took up deuterium, right? Whereas this population here is lighter, has lower mass, meaning it did not take up deuterium. And what you'll actually see is that if you repeat this experiment at different times, this population will go down and this one will go up as that part of the protein starts to fold and resists the process of exchange. And you can analyze not just this yellow-red region, but you can do the same analysis for pretty much every region of the protein, for at least for a well-behaved protein. And you can learn the sequence with which different parts of the protein kind of fold themselves. So it would be great if we could then take the same experiment and do it not for proteins in solution, but proteins when they are being translated on the ribosome. But this is now a much more challenging experiment for a couple different reasons. And so I'm going to highlight what are some of the challenges that we face and how I've resolved them. One of them is that you now have not one dynamic process, but 2 dynamic processes. You have both folding, which is how as the protein gets longer and longer, it starts to acquire all sorts of shapes. And then translation, meaning the fact that the protein is actually getting longer and longer. And so if you have a solution with a bunch of ribosomes within it, how do you kind of take care so that you know that at a given time, the ribosomes have, say, advanced to some degree, whereas at a later time, they're later on? Because if you don't take care of that, the signal you get will be very complicated because every ribosome might be in a different place. And so I've come up with a way of doing this, which is called solving this, which is a synchronized in vitro translation reaction. So it turns out that there's been some really beautiful work that shows that you could actually take the ribosome out of the cell and add to it all of the factors that it needs in order to translate a protein for you in the test tube. So you can have a test tube and within it, your ribosomes will make protein, even though that's not even the cell, but it's sort of a simplification, a reproduction of the cellular environment. And I've come up with a modification of this particular approach where the main change I make is I ensure that this translation is synchronized. It's like you have a lot of swimmers representing the ribosomes that they all start at the beginning of the pool, and then when you want them to start swimming, they more or less swim at the same rate as each other. And the way that it works at a technical level is I pre-initiate my ribosomes on the mRNA by omitting all amino acids except methionine, and then when I want to start the translation, I give it all the amino acids that it needs in order to translate. And you see, for instance, on a gel, larger and larger species. The other challenge is, as I mentioned, the way you figure out what parts of the protein took up deuterium and what parts didn't is you analyze them on the mass spectrometer, right? But now you're not just going to see peptides from this little protein here, right? Because everything in the system is a protein or has proteins, right? The ribosome has many proteins. There are other auxiliary elongation factors that help the ribosome advance. And so this is, in English, we have the analogy needle in the haystack. No sé cómo se dice algo parecido. Like, okay, entonces, yeah, so how do you solve this problem? And so what I've come up with an approach where what I do is I translate a protein that has an N-terminal ABI tag. An ABI tag is a sequence to which a biotin can be covalently attached by the enzyme BRA, a bacterial biotin ligase. And so what I do is I perform the synchronized translation and I add to it a BRA of this particular type of gene. And so the nascent chain gets co-translationally biotinylated. And for those who don't know about biotin, it's this molecule that has an amazing property that it can stick very strongly to another protein called streptavidin. It's an extremely, extremely strong interaction. So what I could do is when I do my labeling of the reaction with deuterium, I can simultaneously add these beads that are coated with streptavidin. And so the nascent protein, as it's coming out from the ribosome, will readily stick to the streptavidin beads. And then, as I mentioned, after you label, and at the same time as you're doing that, you're labeling the hydrogen, you're exchanging them with deuterium, the ones that are unfolded, right? And then I want to stop the reaction. So what I could do is I can acidify the sample, like I mentioned before, that stops the exchange of hydrogen to deuterium. But because I acidify, it has the second effect that it also causes translation to stop and it causes the ribosomes to fall apart, right? So all I have now bound to the beads is my nascent chain. And I can then add to it protease directly to the beads and it digests them into peptides, which I can then inject into mass spec. So it's a lot of detail, but the idea is that this is sort of an approach that I use to solve some of the complications for doing this hydrogen deuterium exchange experiment now on the ribosome during translation. And what I'm going to show you now, I'll show you a little bit of data that tells you we can actually see how different parts of the protein start to fold as they're being translated. So this protein here is halotag. You might remember I mentioned it briefly before. It's a protein that's used, probably people know about it for fluorescence labeling in cells. And it's also an interesting system, especially for us, because we know that it folds better when it is translated versus when it is unfolded and refolded, basically. Meaning something about the translation biases how it folds. So what I've shown here, I'm pointing to two different parts on the three-dimensional structure of the protein. They're very different in where they are in the sequence. This blue one is at the N-terminus, meaning it's one of the very first things that gets made. And then this orange one is at the end, at the C-terminus. And so what I'm showing here are two different sets of mass spectra corresponding respectively to these two regions. So it's a lot of information, but let's look at it one at a time. So these blue plots tell you something about what's going on with this blue region, right? Whether or not it is folded. And what you can see is that at 6 minutes, this spectrum is relatively to the right, compared to what happens to it later at least. And so the fact that it's to the right means that it actually takes up deuterium. So at 6 minutes of translation, it hasn't started to fold yet. In contrast, at 15 minutes, you actually see a change in the shape of the mass spectrum. And it now gets well fit to a combination of this unfolded population and then the small population that is now protected, right? Meaning it's lighter. And then by 4 hours, all you see is this lighter population. So you can actually watch how this region folds. You can also watch for this orange region. At the beginning, you don't even see it, right? Which makes sense because the ribosome just hasn't gotten there yet. You haven't translated it yet. But then starting at 15 minutes, you actually start to again see both populations and then at 4 hours. And the way we know, by the way, where the ribosome is we could actually take these same samples at the same time points and run an anti-biotin Western blot on them to sort of monitor the progress of the reaction. And you can see formation of larger and larger species. And only at 15 minutes, you see this big band here, which is the full-length protein. So that's how we say how translation is kind of happening. And this is just another representation of this exact same data, but with a few more time points, basically. Where what I've shown here is just, I looked at these spectra and I asked what fraction of the population gets fit to this more protected peak, right? Saying, in other words, what fraction is folded, what fraction of the molecules are folded at that time. And same for the orange one. And so you can see how, at early times, before translation is complete, this region is not folded, and then both of them start to fold after translation is complete. And I'm only showing you 2 peptides here, just to make the plot a little easier to watch. But it turns out that if I make the same plot for all the regions of the protein, I actually see pretty similar trend as this, where early on in translation, there's no folding, and it's only after translation is complete that this that all these regions start to fold. So halo tag does not actually fold until translation is complete. And so one question we then asked, we're kind of curious about that. We were like, is this because halo tag is not capable of folding during translation, or is it just because translation maybe is too fast and doesn't give time for the halo tag to fold, right? Again, to use the analogy, what we're kind of seeing, it's like we're seeing that the ball still goes to Santiago, but can it maybe not, but it could have maybe gone to a canyon. Did it not go to the canyon because the wind was not fast enough to make quick erosion, or maybe there was just no canyon at all, right? So this is a sort of question we were trying to understand. And the way that we can answer this question is we can do a slightly different type of experiment, where now, instead of actively translating the protein, you artificially generate constructs where you tell the ribosome to stop at specific locations. So for example, we tell the ribosome, make only 96 amino acids, or make only 150 amino acids for all these different sizes. The way you do this at the technical level is you can give it an mRNA without a stop codon, and the ribosome will just reach the end of the mRNA and stall there. And so I did that experiment where I generated these what are called stalled nascent chains, and I gave them a very long time to fold. I gave them 4 hours, so that if they were capable of folding, we should see that. And what I'm plotting here is the same type of information as before, but now the x-axis, instead of time, it's length. And we're plotting at equilibrium the probability that these different constructs are folded. And what you see is that halo tag is more than capable of folding co-translationally if it were given enough time to do so. So you see these regions, the probability of folding goes up with increasing translation rate. But of course, as we showed before, for all these different regions, no folding actually happens until after translation is complete. And so this tells you that actually the reason we don't see folding is due to a non-equilibrium effect, right? Non-equilibrium having to do with these coupling of timescales not quite lining up. And this, I think one of the takeaways is it points to the importance of studying this process with experiments that do active translation. Because it turns out in this field of co-translational folding, the more common way to study folding on the ribosome is this type of experiment, where you generate stalled nascent chains. It's just a simpler experiment to do. You don't have to worry about synchronization and all these other issues. And oftentimes people will see length-dependent folding like I see here. But if we had just done that, we would have thought that halo tag has a co-translational folding pathway, but it actually doesn't. And so it really points to the importance of methods that can capture sort of non-equilibrium processes. And so here's one thing that we haven't resolved though. I told you at the beginning that halo tag is a protein that benefits from translation and achieves more folding like that than when you unfold it and refold it. Yet I also just showed you that halo tag doesn't actually fold during real-time translation. So what's going on? It turns out that there is a kind of hysteresis effect that happens, where the fact that halo tag was just translated somehow then makes the protein fold in a different way once it's released from the ribosome versus when you unfold it and refold it. And here's how we see that. So I've kind of summarized the many different mass spectra from before into these cartoon formats, where what I'm showing here is I'm sort of showing the sequence with which different parts of the protein start to fold or get protected after they're released from the ribosome. And the parts that I've color-coded are the first ones to fold, and then by the end, I've color-coded everything, meaning eventually everything folds. So we see that this part of the protein and these parts are actually the first ones to fold. If you then do the different experiment, the experiment in the style of Christian Anfinsen, where you unfold and refold the protein, there's also what we call a folding intermediate, so some parts fold first. But the parts that fold first are very different here in this simplified experiment versus in the translational experiment. And so what's quite interesting about this is we know that part of the reason why halotag does not fold very well in isolation is because it's prone to aggregation. And there's evidence that this is actually the intermediate that aggregates. So by avoiding this intermediate and instead folding starting from the C terminus, you get around this. So there's some sort of hysteresis. Somehow translation affects the folding pathway. We don't fully understand why that is. More likely than not, what's happening is that during translation, some amount of folding happens, but that structure is too weak to produce a very observable amount of protection from deuterium. So I actually have a student now who's helping me run simulations with the algorithm I'll tell you about in a little bit to see if we can kind of try and explain this observation. So, okay, we talked about halotag, and we saw that halotag doesn't actually fold to an appreciable degree until after translation is complete. Even though it is capable of it, but due to the finite rate of translation, it doesn't have time to. Is this result general, or maybe are there other proteins that do actually fold even before the completion of translation? It turns out that there actually are some that do. And so I'll summarize results from one other protein that I've been looking at, which is a protein from the Timbarrel family. So Timbarrels, as some of you might know, it's one type of protein structure. Proteins can be categorized into different fold families depending on the shape of the different atoms within the protein. And yeah, by the way, for those who maybe are not so familiar with how we look at these protein structures, these are kind of ribbons and arrows that basically show if you take the chain of amino acids and sort of arrange it in space, it's ways of sort of monitoring how the chain progresses. So you see that some of them are arrows, meaning that's the direction in which sort of you go from the beginning to the end of the chain. And then the parts that are sort of squiggly like this are what are called alpha helices, so they have local structure. And then when you have these arrows next to each other, this is a type of longer range structure. It's just sort of one representation of the structure. So what we're going to look at is we're going to look at a part of the Timbarrel that is well folded in the native state, meaning that when the protein is correctly folded, we know that it's very much sort of very buried, very much at the core of the protein. And we're going to look at what happens to that region of the protein at different points during translation. And as a control, which I also did before, but I didn't show you, you can also do a control in which you can artificially unfold the protein and ask how much deuterium does that region of the protein take up when it's unfolded. And this is what that spectrum looks like. And what we see is that at 7-8 minutes, which from gel we know is before translation is complete, the mass spectrum is already shifted to the left relative to this unfolded population, meaning that even before translation is finished, that part of the protein already starts to fold. It's not quite as folded as in the native state when the protein is correctly structured, because the spectrum now is even more shifted to the left. But some intermediate amount of folding is actually observed on the ribosome, and I see it for a few other regions as well. As a control, let's look at a part of the protein that's very disordered, even in the native state, meaning in the native state, it's just exposed to water, it doesn't really form much structure. And what we actually see in that case is that the mass spectra looks pretty much the same as the unfolded protein the whole time. So that tells you that we're seeing something that's region-specific, that's structure-specific. So we attribute that to mean that the protein actually folds co-translationally, as it's even before translation is complete. And so we can summarize, again, I showed you just two peptides, two regions of the protein. But if we do this experiment, if we compile the information we get from all of the different regions of the protein, we can make a little bit of a cartoon of how this protein folds co-translation. So it's like those very first cartoons I showed, but now the nice thing is this cartoon is actually based on data. And so the way that I've color-coded this is at the different stages in folding, I've indicated those parts of the protein that are partially folded with these stripe colors like this, whereas parts that are fully folded, like at the end, I've indicated in the solid color. And you can see that there's a sequence where these beta strands form first, and then the other ones kind of dock on, and then by the end, everything is folded. The details are a little bit less important than kind of the fact that you can actually resolve this hierarchical series of co-translational intermediates. And again, it's during real-time translation. So let me conclude oh wow, it turned more blurry than I thought. But let me conclude the first part of this talk, which is the fact that we can now actually see how a nascent protein folds as it's being actively translated, something we were not able to do before. And what are some of the lessons we learned? We learned that actively translating proteins show very rich non-equilibrium behaviors that cannot be captured on experiments that assume that everything reaches equilibrium or that measure things. 10 minutos?
Yes.
Okay, muy bien, thank you. And so we see that with actively translating proteins, you need approaches that capture non-equilibrium effects like this. We also see that different proteins have diverse co-translational folding mechanisms. I've shown you two, but I also have some data for a third that I'm kind of still analyzing, and it seems to be somewhere in between these two examples. And so we see that folding pathways can be diverse on the ribosome. They might resemble, in some cases, so which I forgot to tell you this, that for this protein, alpha-tryptophan synthase, this folding mechanism is actually pretty similar to if you just take the protein in solution, purified protein, unfold it and refold it, which is very different than halo tag. And so in some cases, doing experiments on these simplified, purified protein will already tell you the correct answer, even if it wasn't co-translation. But then other cases, the ribosome causes folding to happen in a very different way. And in those cases, those differences can actually be very important in affecting the yield, how much of the proteins end up folded correctly. And so we also have, since we have this method, it's pretty generalizable. And so we can then take it and apply it to all types of interesting proteins, including ones that have been implicated in misfolding disease. And you can ask whether any of these misfolding defects might already originate on the ribosome. And that's something I'm very excited to take this direction in. So I'll just give a little bit of more of a summary overview. This work that I'll talk about now is published in a couple of different places, but the majority will be this last publication. So you can read about it in more detail with a limited time. So the idea is that simulation, these experiments like hydrogen-deuterium exchange, they're very high resolution. So you can see what's happening in many parts of the protein, but nonetheless, they are not structural experiments. So you don't actually know where all the atoms are during these different stages of folding. And I'll tell you a little story about how with simulation, you can actually fill in that detail, all atom simulation. And so I'm telling you a little summary of some method development work I did in my PhD, where computational methods have always played a very big role in this field of protein folding. But really, there are not many methods that can actually, in a comprehensive way, tell you what is the mechanism, how a protein folds, and that can actually capture the different stages in the folding process. You have methods that make some simplifications, and you have AlphaFold that very quickly but rapidly predicts a native state. But being able to simulate the different steps in protein folding with a reasonable amount of time so that the simulation doesn't take decades to run is very hard to do. And so what I developed an approach where you can actually get around this, and it's a Monte Carlo based approach. I can talk a little bit in more detail to anyone who's interested, but the basic idea behind it is you actually simulate protein unfolding instead of refolding, and you add in what's called enhanced sampling. It's a way of sort of affecting how the simulation works. And then if you set it up in a correct way, you could actually then learn about the opposite process using an idea from statistical physics called detailed balance. And I know this is very fast, but I can talk about it more later if somebody's interested. The main takeaways is that this is a way of figuring out how a protein folds. You now do it on the computer. You watch the protein sort of get its shape one step at a time, and you can watch at every atom. And this does something that accounts for what are called non-native interactions, meaning that it can actually, even if maybe it's possible that when this chain of amino acids is starting to fold, maybe it collapses first in a way that brings together amino acids that you would not expect if you know how the protein eventually looks like. It might sort of it's like if I'm trying to go from here to the desk, instead of going straight to the desk, maybe I first go grab my water and then I go back and then I go to the desk. And this algorithm can sort of predict how that might happen. And it's a pretty generalizable method. And so what we did, we had a collaboration between my PhD lab, Shaknovich lab, and the lab of Marina Rodnina in Germany, in Göttingen, where we wanted to understand for this one protein, which we know folds co-translationally through a sequence of intermediates, what those intermediates might look like. And that's known from some PAD experiments, as well as an experiment called SECAM, which I can tell you guys more about if you're interested. But we didn't really know what those stages in co-translational folding look like at the atomistic level. And so we wondered if with simulation, we could actually learn about those different stages in the folding process. And so the way we did that is we used this algorithm to run simulations of this protein at many different sizes, mimicking the different stages in the translation process. And what you can do, you can analyze those simulations by making what are called contact maps, which tell you with what probability different pairs of amino acids make contact with each other or come close together. And on the bottom left are what are called non-native contacts, which are ones that you wouldn't expect. And then on the top right, it's ones that you would expect. And you can do this kind of at many different lengths, and you can then ask at each length what fraction of the contacts are native versus non-native. And you see, this was kind of known from experiment, but we didn't know it in detail, that this protein during translation will transition from a non-native state to a more native-like state. So it is as if, this protein, if it tries to get to the desk, it goes to the water first and then goes to the desk, right? And so we did some other characterization where we showed that these non-native intermediates are actually pretty compact, have a sort of compact radius of gyration, so they might be able to fit in the exit tunnel. There's a lot of analysis we did. I can tell you guys about it if you're interested. But one of the most striking things we found is that there are some amino acids, which if you look at where they are in space, when the protein is completely made, those amino acids are sort of facing outwards. They're exposed to the solvent around them. But when you actually look at where those amino acids are in middle stages in translation, they're actually facing the inside. They form what's called a non-native hydrophobic core. And that's not general. It turns out that there are other hydrophobic amino acids that don't play quite as an important role, that actually do stabilize the native state, but are not as important at intermediate lengths. And so you see that one of the main predictions of the simulation is that these intermediates are stabilized by a very specific set of amino acids that are not so important in providing stability of the full-length protein, because they're out here, but they are important at these intermediate stages in translation. And we were able to use the simulation to generate an experimental prediction, which is what would happen if you mutate these residues. It turns out they have an approach called PET fluorescence correlation spectroscopy, PET-FCS, where they can measure the fluctuations in the distance between two amino acids in the protein as a proxy for stability. And we asked here, at these different stages in translation, how much would the flexibility of these regions change if you mutated those two groups of residues, right? And prediction was that for that first group, you would destabilize the protein early on in translation, but not as much later. But for that second group of amino acids, you have still a little bit, you destabilize it at the beginning, but relatively the effect is more later on in translation, right? And it turns out that is exactly what they saw. So when they did the PET-FCS experiment, they found that this first group of mutations has predominantly an effect during translation, whereas the second group has an effect on the stability at the end, which is very exciting. And they did quite a bit of characterization, also showing how these early intermediates might be affected by the presence of the chaperone trigger factor. They did a very interesting characterization, and a lot of the details were actually consistent with simulation. And I'm happy to talk more about it or point you to this publication. But the main takeaway, big picture, is that with this simulation approach, we can now, at an atomistic level, we can predict when the protein is trying to fold in its native shape, including on the ribosome. We can predict how where the different amino acids actually are in these different stages, and that the predictions are experimentally consistent. And so this kind of suggests, and I also did a little bit of work earlier in my PhD showing how co-translational folding can help certain proteins actually avoid misfolded traps that happen later on in translation, and how rare codons might actually be important in giving time for this process. And this you can read more about in this paper here. So the main idea is you can actually gain a lot by combining experiment with simulation, right? With experiment, you can sort of see what's actually happening at quite a high level of resolution, especially now with HDX-MS, but it's still not an atomistically resolved technique. But we also have an atomistic method, which, by the way, the predictions from here could not be captured by AlphaFold, because it turns out that if you take proteins of different lengths and put it into AlphaFold to predict how it might fold co-translationally, what it will tell you is that at every length in translation, it will adopt a native-like state, right? But we see very clearly that that's not the case, and so you need physics-based approaches to be able to make these types of predictions, or maybe more sophisticated machine learning, but that would need to be trained, of course, on new experiments or new simulations that we're only starting to have now. And so you can see how it's a very powerful combination to have the two, and I'm also excited, since I've done some work on both, now actually combining my own experiments with my own simulations. And so stay tuned, especially for Halo Tag. I think we'll be able to learn a lot with that approach. And also just some future directions, again, sort of zooming out why this is important, where this work will go in the future. We can sort of, use this, we have these methods, both simulation and experiment, that we can ask a lot of interesting questions, like how is it that in translation, when the landscape of a protein is always changing, how that might affect its ability to fold, how evolution has selected for that with codon usage. We can then expand these experimental approaches to include chaperones, to include interaction partners. So, because there are some proteins that don't work alone, they actually make interactions with other proteins. And sometimes there's evidence that those interactions start when one of those proteins is first made on the ribosome, the partners will come and join it. And so it would be interesting to watch that and watch what are called chaperones, which help facilitate folding in some cases. And as I mentioned before, it would be then very interesting to see whether in cases of misfolding disease, like ALS and some others, whether those defects in folding might already originate co-translationally on the right. And with that, I would like to thank a number of people. So first, my funding. I'm especially honored to have been a part of the Jane Coffin Childs postdoctoral fellowship the last few years. I have some very talented undergrad that have helped in distinct stages of this project. This group here is from the Marina Rodina's lab in Germany. It was a thrill to collaborate with them and get their support on this simulation project. I'd like to thank Eugene Chaknovich and also a number of his postdocs and students, especially Will Jacobs, who trained me in the simulation. I'd like to really thank my postdoctoral advisors, as well as Ito's, Susan Marquesi and Carlos Bustamante, and the whole lab. They've been such a really intellectually stimulating environment. It's one of the most, they're two of the most rigorous environments I've ever been in. They're like, you know, the group meetings, you remember, will last 3 hours, right? Sometimes, especially Carlos's, will last 3 hours. Susan's could also last, but she always cuts it off at 2 hours. But because every little thing you show, people will take it apart, will, you know, find a way to make it better, which, you know, can be hard, but you really, that's how I grow as a scientist. And so, muchas gracias también a Ito por la muy cálida invitación y cualquier pregunta.
Gracias, Amir. Ahora tenemos tiempo para preguntas de online o en público. Puede ser en inglés. Ahora en castellano, la persona de inglés se fue ahora. Por eso puede ser en castellano. En castellano. Sí, sí. La tres primero.
Quiero preguntar, ¿cuánto es el tiempo de simulación necesario para tener una predicción de folding eficiente?
Depende del tamaño de la proteína que estés simulando. O sea, esta proteína GMK es relativamente pequeña. En el tamaño completo, al final de la traducción, es 79 aminoácidos. Y para cada tamaño de cadena que nos interesa, requiere como una semana más o menos de wall time, de tiempo de pared. Pero también bastantes CPUs, o sea, no es barato. Es un método que sí es un poco caro, pero relativamente en comparación a otros es bastante eficiente.
El tiempo de simulación es un nanosegundo.
Ok, buena pregunta. Esta simulación es un tipo de simulación llamada Monte Carlo simulación, o sea, no te mide el tiempo en tiempo real. Es un poco complicado. Lo que hace es como una manera de experimentar diferentes estructuras en el terreno energético de la proteína. Es un poco difícil decir a qué corresponde en tiempo real. Para saber eso hay que calibrarlo contra simulaciones, pero te podría decir que te permite simular procesos que tomarían cabo en escalas temporales de, en algunos casos, segundos a minutos, a pesar de que no te esté literalmente simulando un segundo de tiempo.
Gracias.
¿Tú? Sí.
Sí, a mí me quedó una duda específicamente de la parte de las proyecciones posibles, la asociación con la enfermedad con Alzheimer.
Ajá.
Porque al final del día, a menos que haya ocurrido una mutación muy, digamos, una mutación espontánea, algo tiene que cambiar entonces en la capacidad o la velocidad quizás de la traducción para que eso ocurra. No sé si es que sabes de algo que podría estar ocurriendo en estos pacientes quizás.
Pues no tiene que ser necesariamente la velocidad. Eso podría suceder si hubieran sustituciones sinónimas. De hecho, en el caso del cystic fibrosis sí se han encontrado que ciertas sustituciones sinónimas causan ciertas variantes de la enfermedad. Pero te podrías imaginar que otros factores involucrados en el plegamiento de co-traduccional también podrían, o post-traduccional, también podrían cambiar, ¿no? Por ejemplo, en el caso del ALS, se sabe que la proteína SOD1 interactúa con varios metales, ¿no? Con el cobre, con el zinc, y si hubieran cambios en el ambiente celular que causara una pérdida de esos iones, eso también podría afectar el plegamiento, ya sea en el ribosoma o después del ribosoma. O sea, pueden haber muchos factores ambientales que también lo causen, no tiene que ser mutaciones en sí.
Hay una pregunta online,
Sí.
Simón, si puede.
Hola, ¿me escuchan?
Sí.
Ya, súper. Tengo un par de preguntas. Bueno, tengo varias, está súper interesante la charla. Quería saber primero si todo esto que uno aprende del plegamiento en dos fases, primero de los residuos hidrofóbicos adentro y los hidrofílicos afuera, y después la formación de estructura terciaria, ¿se sigue cumpliendo en este caso o el plegamiento es más rápido o hay un reordenamiento distinto?
Sí, buena pregunta. Una pregunta interesante, no había pensado tanto en esos términos. Bueno, lo que medimos en este experimento realmente es difícil contestar esa pregunta porque este experimento no nos dice mucho de colapso hidrofóbico. Lo que te cuenta es de formación de estructura secundaria estable. Yo apuesto que tu mecanismo, o el mecanismo que me contaste aún es cierto, no más que esas primeras etapas no las podemos realmente detectar. De hecho, yo creo que lo que pasa con la proteína Hydrogen debe haber algún tipo de colapso que sucede hidrofóbico que sucede en el ribosoma que no podemos detectar precisamente por el hecho de que esto no introduce suficiente protección a los ámidos para poder detectarlo. Pero según la simulación, la segunda parte de la plática, ahí sí podemos ver evidencia de casos en la cual la proteína está colapsada, pero no necesariamente le vemos estructura estable secundaria.
Ok, ya. Y la otra pregunta es que estaba pensando también si las simulaciones se podrían evitar y hacer en AlphaFold, pero dijiste que no.
Yo creo que no, sí.
Sí, no, tiene sentido, tiene sentido. A lo mejor, no sé, un Rosetta podría salir mejor, pero me pregunto yo si a lo mejor puede ser que un análisis de la estructura secundaria por pedazos se podría hacer que hay muchos más métodos de estructura secundaria tal vez que son menos tendenciosos que AlphaFold. Y entonces
Ajá.
la pregunta sería, ¿hay diferencias notables en la estructura secundaria que se puedan detectar a través de la... o son más de reordenamiento de hélices y de dominio?
Para asegurarme que entiende, con la estructura secundaria, ¿qué sería lo que haría con esa información de la estructura secundaria? ¿Cómo
O sea,
usaría eso para predecir el plegamiento?
lo que pensaría yo sería si hago este análisis de ir plegando de pedacitos, ¿cierto? De partir con un dominio, después
Ujú,
perdón, con otro segmento, si ver si
ujú.
efectivamente las estructuras secundarias difieren mucho de las nativas y de ahí poder descartar si es que, o reforzar, digamos, esto antes de realizar las simulaciones. O a lo mejor guiar un poco más las simulaciones.
Bueno, si una simulación, ¿cómo sabría que, o sea, dices del experimento, ¿cómo lo sabría si son diferentes a las nativas? ¿Con experimento?
Ah, ya, bueno, no. Estaba pensando que uno siempre sabe cuál es la nativa. Eso es lo que...
Sí, sí, o sea, sí, lo que te dice la simulación realmente es esto de la estructura terciaria, no solamente la secundaria. Yo creo que
Ya.
esa es una de las grandes ventajas. Y obviamente si hay un modelo muy sencillo donde tenemos razón pensar que nada complicado sucede, quizá algo así es suficiente, pero en un caso como que esta proteína se sabía que tenía un intermedio no nativo, entonces algo sí hacía falta hacer algo un poco más sofisticado.
Ya, ok. Gracias.
Sí, sí, por favor.
Yo quería entender si es que las enfermedades tienen problemas de folding, ¿no? Yo me imagino que eventualmente una vez que ustedes logren comprender en qué momento y por qué causa se produce el misfolding,
Ujú,
entonces podría haber una intervención
sí.
que evitara
Ujú,
el misfolding,
ujú.
¿cierto? Ahora, el misfolding se produce por causas que están tratando de determinar, pero ¿la presencia de aminoácidos es correcta o podría producirse también porque no están los tipos y la
Bien.
cantidad de aminoácidos presentes?
Bien, buena pregunta. Ambas pueden ser cierto. Entonces, por ejemplo, enfermedades como Alzheimer's se categorizan en dos tipos. No hay Alzheimer's familiar y Alzheimer's esporádico, ¿no? El caso familiar es exactamente ese mecanismo que propusiste que en ciertos casos mutaciones causan que la composición de aminoácidos no sea correcta. Y de cierta manera este tipo es un poco más fácil de entenderlo. Pero también hay casos esporádicos que no se entienden realmente, pero se cree que tienen más bien que ver con factores ambientales. Y ahí sí que algo más complejo, algo que tiene que ver con el ambiente en la célula, con el procesamiento de energía, puede haber miles de factores. Y ahí sí es importante entender qué es lo que está pasando para poder intervenir. Y efectivamente han habido casos, por ejemplo, la enfermedad en inglés se llama transthyretin amyloidosis, un tipo de amiloidosis que afecta al corazón, los riñones y otras partes, que un científico ¿Saben de quién hablo? Un científico en Scripps en San Diego que hizo un estudio muy detallado del plegamiento de esta proteína en caso de un paciente sano y luego identificó un defecto en una etapa intermedia del plegamiento que sucede en pacientes con esta enfermedad y desarrolló una molécula farmacéutica que corrige a ese proceso de plegamiento. Y esta molécula ha extendido la vida de estos pacientes 20 años, hasta que hay gente con esta enfermedad que ahora muere por otras causas. Un milagro. Y bueno, la meta sería poder hacer este tipo de intervenciones.
Amir, una pregunta.
Sí.
Recuerdo, ya, entonces si entiendo bien, la proteína espera traducirse completa
La gelota.
para después empezar a plegarse.
Sí.
Y tú ves que las zonas de plegamiento son diferentes, tanto la proteína que está con el ribosoma como la proteína diferente. La
Sí.
proteína sin ribosoma.
Sí, la que se renaturalización. Sí.
También recuerdo un trabajo del grupo de Carlos Bustamante, Christian Kaiser, Daniel Goldman,
Ajá.
que ellos hacían pinzas ópticas
Ajá.
y determinaron que el ribosoma funcionaba como chaperona
Sí.
porque había una zona de carga a
Sí.
la salida del exit tunnel.
Ujú.
Ok. ¿Cómo se relaciona esa carga que tiene alrededor del exit tunnel con el orden de plegamiento en el ribosoma? Probables cantidades diferentes de sal, de fuerza iónica, para ver si con mucha fuerza iónica ya se pliega como afuera del ribosoma. ¿Me entiendes?
Sí, buena pregunta. No lo he probado. Es un poco difícil porque estos sistemas de traducción in vitro son tan optimizados, ¿no? Requieren cierta cantidad de sal, ciertas condiciones para traducir efectivamente. Y me preocupo que hacerle un cambio a la cantidad de sal también va a afectar no solamente quizás las interacciones electrostáticas, pero también quizás la velocidad de traducción, miles de otras cosas. Pero entiendo lo que sugieres. Tendría que pensar cómo hacerlo de tal manera de no afectar. Quizás simplemente en cadenas arrestadas, aunque ahí pierdes el efecto fuera de equilibrio, ¿no? Entonces, tendría que pensar cómo hacerlo.
Claro, porque recuerdo eso que era como chaperón, ¿no?
Sí,
El
sí.
efecto.
Sí, lo que recuerdo también es el trabajo de Lisa Alexander, porque lo que Lisa Alexander mostró es que la traducción activa introduce algún tipo de cambio en el camino, en la trayectoria del plegamiento que no se podría predecir en cadenas arrestadas. Y esto parece ser parecido, no sé si idéntico, pero algo parecido.
¿Tenemos tiempo para una última pregunta? Sí, por favor.
My English.
Yeah, sure.
Not that English don't know anything. Excuse me. Is there a third logical possibility to the other two that were mentioned before, which is the non-native state will have some functional role such that you will have diseases that are in which you attain the native state, but the folding will be abnormal.
Mhm.
And it's these changes in the online folding process that could functionally result in disease to other, say, if the non-native state has a role in other, it's necessary for some other processes.
So you're saying that you're asking if like some change to a short-lived intermediate in the middle of folding, maybe that intermediate then recognizes yes. So actually, I think this is a good question for your lab because, things like co-translational intermediates can be recognized by secretion machinery and stuff like that, right? And then get sent to the membrane. So I definitely think so. Maybe I think you guys can better answer a specific case or something like.
Yeah, you're right. It's very important, the intermediate for functionality. And many people just think about the folded structure is important, but sometimes even the unfolded structure is important for function. There are some proteins are called intrinsically disordered protein that those proteins function in your body unfolded, and then one part of your protein is folded, native structure, but the half of your protein or a third of your protein is intrinsically disordered. And that is extremely important to function because some of the intrinsically disordered protein create, they call phase separation.
Yeah.
They allow to separate from the other protein. And you keep that protein isolated until you need it. And when this protein know when you need it, for example, if you eat something or you create some molecule of too much AMP, a molecule in your body, then that part eventually will fold. and then we'll attack or we'll, you know, your question is really good.
Yeah, I was telling, we were having, ¿cómo se llama donde cenamos el otro día, el restaurante italiano? No, el otro, pero bueno.
¿En Chile?
Sí,
¿Golfo de Nápoles?
No, entonces les estaba explicando a mi familia, estaba mezclando aceite con vinagre y se formaron las gotitas, se les mostré, esto pasa en la célula también, pero
Sí,
no,
se llama con las proteínas.
tiene confirmación también. No, incluso el amyloid beta, la que se agrega en el Alzheimer's, es también un IDP, no lo mencioné, intrinsically disorder, no lo mencioné, pero pues sí, son
Súper bueno.
Ujú.
Para finalizar, nuestra coordinadora estratégica de Relaciones Internacionales nos trae un regalo,
Gracias.
porque Chile es un país que tiene mucho cobre, así que vas a ver el regalo directamente de las minas de Chuquicamata.
Ay, qué lindo,
Un lápiz de cobre.
Siempre se me pierden las plumas, pero esta no. Gracias, muchas gracias.
Un aplauso. Y ahora los invitamos a unas galletas afuera. Gracias, la gente online, Simón, Mauricio, Francesca.
Chao, Fran.
Gracias.
Gracias.
Chao, Amir.
Chao, nos
Excelente
vemos.
charla.
Chao, Francesca. Saludos.
Saludos, un abrazo a todos.
Un abrazo.
No. Gracias.